Okay, so now I'm going to talk about Alzheimer disease, the pathophysiology of Alzheimer disease. So Alzheimer disease, it is a neurodegenerative disease. That means there is a loss of neuron, especially in the cortex of the cerebrum and hippocampus. So to talk about Alzheimer disease, first we have to think of a nerve cell. So this is how a nerve cell looks like. Okay, dendrite. So this is a bipolar nerve cell that can be found in the CNS. So from here, we are going to talk about the hypothesis being suggested. So one of the hypotheses is that the presence of the plaques and tangles, which is going to cause the development of Alzheimer disease. So we have plaques and tangles. Okay, now we'll focus on plaques first. So to talk about plaques, we are going to focus on the cell membrane. Okay, so this is how the cell membrane looks like. Phospholipid bilayer, and in between this cell membrane we actually have a protein. We call it as APP protein. This is a transmembrane protein. You guys can just draw it in such a way. So you just follow this line. So this is the head, right? So you just follow this line. So draw a line over here because they have two subunits. Okay. And then this is the transmembrane protein. In this case, we call it as APP, amyloid precursor protein. Okay, so this protein is actually helping the neuron to grow and also to repair. Since APP is a protein, it is going to be used and then broken down and then being recycled. So who is going to break down the protein? So it is the enzyme. So in this case, we have secretase as the enzyme to break down this particular protein. And we have three types of secretase, name it beta, alpha, and gamma. Okay, so gamma is a very important protein because they can be found anywhere and they are going to work with any of the conditions. They are going to work with any of the secretase in any of the conditions. So this is a very important enzyme, gamma secretase. And in a normal condition, we have alpha secretase and gamma secretase to work together. So how are you going to memorize these three things is that you can just think of it as BAQ. Our exam questions, BAQ. So just think that this Q is actually gamma. So BAQ, it is arranged in such a way. And now we'll talk about the APP protein. So in normal case, still we have the APP protein. If this is the cell membrane, what you have to do is this head, we have to match with the line. Okay, it looks like this. And then arrange these enzymes back to here. So we have beta, alpha, and gamma. So in normal condition, we have alpha and gamma. Alpha and gamma. In normal condition, we have alpha and gamma. So what this enzyme works is that, just now I mentioned about, it is the enzyme that is going to break down the protein. So by how we are going to break down the protein? Protein is made up of amino acid. So we are going to cut the peptide bond. So we are going to cut this region. Also, we are going to cut this region. So this is corresponds to the place where the cutting point is located. So beta is going to cut here, alpha is going to cut here, and gamma is going to cut here. And in this condition, in normal condition, we are going to talk only about alpha and gamma secretase. So they are going to cut over this region. So what they form is these two molecules. So these two molecules is actually soluble. So it will be fine for it to be excreted, for them to be excreted out of the body. So there's no issue over here. So this is what happens in normal condition, but what happens in Alzheimer's disease? So just now we mentioned about the normal enzyme is the alpha and gamma. And in Alzheimer's disease, we have the enzyme. Just now I mentioned about also the gamma secretase is going to present in any of the conditions. So we have beta secretase and gamma secretase. Okay. So where it is going to cut is that, again, we are going to arrange it in such a way. Okay. Put all the enzymes here, beta and alpha and gamma. So beta, just now I mentioned about Alzheimer's disease, we are going to talk about beta and gamma. Gamma is going to present no matter what condition. So gamma is going to cut over this region, just like what happens over here. And then beta, we are not going to talk about alpha. And then beta, it is going to cut over this region. So what is the end product? It's going to form this thing and this thing, right? So this molecule, obviously, it is actually larger than this, right? Because initially we actually cut down this thing and this thing. So this molecule is actually smaller. This one, we are going to cut over this area and this area. So the whole block, the whole subunit is going to come out. So this is so large that it is insoluble, just like if you are going to compare with the rock sugar and sugars in the granule form. So rock sugar hardly to be soluble, right? So this is insoluble. So what happens here is that these insoluble particles, they are going to clump together with other protein. In this case, this insoluble protein, we call it as beta amyloid protein or monomer. The reason why it is called as beta amyloid is because they are precursor. They are amyloid precursor protein. And then beta is because they are formed just because of the beta secretase. So the name beta amyloid monomer. And they are going to clump together with other monomer to form the beta amyloid plaques, to form the beta amyloid plaques. So this is going to clump together just like the cholesterol plaques, right? So the oxidized LDL is going to clump together with the cholesterol, and the more cholesterol being accumulated, the larger the plaques, something like that. So we have beta amyloid plaques. Just now I mentioned about this particular protein, it is going to located in the cell membrane. So by having the insoluble protein, that means that particular protein is going to be located outside over here. So it is insoluble, and then they are going to clump together over here. Let's say this is the plaques. And what happens now is that because of the presence of the plaques, normally they are going to have synapse with another neuron. So this neuron is going to form synapse. Supposingly, it is going to form synapse with another neuron. But what happens is that we have, in extracellular, we have this plaques. The presence of this plaque, it is going to stop the transmission of the nerve impulses. So when there is no transmission of nerve impulses, there is no relay of the messages between the neurons. That means no memory is being formed, especially in the region of hippocampus. So no short-term memory, and after that, because of no short-term memory, it is not going to be transferred to the cerebral cortex to sort as a long-term memory. So this is what happens in plaques. And now we'll look at what happens in tangles. Okay, so in the conditions of the tangles, in the case of tangles, so again, just now we mentioned about the cell membrane. Now we are going to focus on the inner cytoplasm. So what is the thing that maintains the structure of the cell? That is the cytoskeleton. And in this case, we are going to talk about the microtubulus, which is one of the components of cytoskeleton. So if let's say this is a cell, we have a lot of microtubulus. Okay, so inside this microtubulus, we are actually having a protein, again a protein. It is called as a tau protein. So the function of this tau protein, it is going to bind to the microtubulus and make sure that it is firm enough to support the structure of the cell, and in this case the neuronal cell. So this is the tau protein. So tau in Malay is like you know or not, like know, K-N-O-W. So tau ke tak tau means you know or not, so tau protein. So okay, in this case tau protein, we have tau protein that is going to help in the maintaining of the structure of the whole cell due to the presence of the cytoskeletons. And in the case of Alzheimer's disease, just now we mentioned about the plaques. So these plaques that is going to be accumulated outside the cell, it stands a very high chance that it is going to trigger changes inside the cell. By how it is going to trigger the changes is that it is going to activate the enzyme called kinase. So what is the function of kinase? Kinase, its function is to phosphorylate. So what happens is that it is going to transfer a phosphate group to this tau protein. So when it transfers it to this tau protein, so this tau protein, like for example, initially it looks like this. Now it looks like this. So what happened? There is a change in the shape of this particular protein. It is just similar to the lock and key mechanism. So when there is a change in the shape of the protein, it will no longer be suitable to fit into this particular microtubulus, so they cannot bind together to make sure that it is firm enough to support the cell. So what happens is that after the phosphorylation of the tau protein, or in this case hyperphosphorylation of the tau protein, so the tau protein is going to free itself out of the tubulus. So you can see there is a lot of tau proteins over here inside the cell. Tau protein. So we can see a lot of tau proteins over here. So this tau protein, same, they are going to, just like the plaques, they are going to accumulate together. But by how they are going to accumulate is that they are going to tangle around with each other. So tau protein, they are going to tangle around with each other. And in this case, we call it as neurofibrillary tangles, which is made up of tau protein. So what happens when we have neurofibrillary tangles? That means the tau protein, it will no longer be there in the microtubulus. So there is nothing to support the cytoskeleton or support the cell.